Hello and welcome everybody to the Biognosis user seminar entitled Exploring Multiplexed Proteomics in Complex Applications with SpectroMine. My name is Lukas Reiter and I'm CTO at Biognosis and I will be your host today. I'm very much excited to have today with me Mikhail Savitsky and Irving as speakers. They will present their research at the forefront of what's possible in proteomics today. To the audience, if you have questions, please share those with us with the Zoom functionality in the Q&A box. We will have a joint Q&A after both presentations in the end, so please let us know of your questions, that it's going to be an interactive Q&A in the end. Our first speaker is going to be Mikhail Savitsky. Mikhail did his PhD in the lab of Roman Zubarev. After his PhD, he held a group leader position at Cellsome in Heidelberg. And then afterwards, he switched back to academia as a group leader at the EMBL in Heidelberg, heading a group, but also the core facility. Mikhail is probably best known for his work in structural proteomics, but also isobaric labeling quantification. And I'm very happy to hand over to Misha now. Misha, the floor is all yours.
Thank you for this very kind introduction, Lucas. So the title of my talk is Multiplexed Proteomics for Understanding Molecular Biology. What I will talk about is indeed a technology that we developed now quite some time ago, which holds a quite prominent place in my lab. It's called thermal protein profiling. It combines the principle of the cellular thermal shift with multiplexed quantitative mass spectrometry. And the concept here is basically that proteins initially was developed to map protein-drug interactions, because proteins, when they interact with the ligand, then become more thermostable and require a high temperature to unfold. So that's been known since the 50s. And so this is utilized here basically in a way that we assess all proteins using relative TMT quantification. So thousands of proteins across the treated and untreated condition, and we record melting curves for these proteins. And then we can compare using bioinformatical computational tools. We can compare these melting curves and we can infer whether thermal stability of a protein changed due to interaction with the ligand. And this would then tell us that there was indeed an interaction between the protein and the ligand or a drug, and this happened inside the living cell. So this is the concept, because we hit intact cells. So that was the state of things in 2014, and during the last years, we've been here at EMBL, we've been working at that quite extensively, and so the technology morphed quite dramatically. And so aside from this figure being more visually appealing, there's actually also some real developments in terms of technology and biology here. And I'll just take you quite quickly, step by step, through what we've done in recent years. So initially, we quite dramatically increased the sensitivity of this technology. This is illustrated here. Basically, by rejiggling how we do these experiments, we were able to, as you see below there's the melting curve of a treated and untreated condition. And you see that the shift in melting points is really, really minute. And we were able to set up technology in such a way that we can actually detect such hits very reliably, as showcased in this heat map above the melting curve. So this is now a significant hit, and indeed a true and validated target of a cancer drug, which is very interesting from many aspects. And this becomes then very exciting, because when you can very sensitively detect small changes in protein thermal stability, this opens the door beyond protein-drug interactions, because indeed any change in protein state, such as protein-protein interaction, protein metabolite interaction, protein DNA interaction, has the potential to change protein thermal stability. So it opens the door to phenotyping the cell and finding out how protein state changes, and specifically those protein state changes which are not reflected in protein expression. So we used that initially to study the eukaryotic cell cycle. We found out a lot of novel, exciting biology about the eukaryotic cell cycle. We then expanded it in a fantastic collaboration with a friend and colleague of mine, Nasus Tsipas, to prokaryotes. And I will talk about this quite a bit more later. So we adapted this technology to prokaryotes. We also elucidated the mode of action of novel potential antibiotics. We expanded this also to mapping protein metabolite interactions in a fantastic collaboration with my former colleagues at Cellsome. We also expanded it to an in vivo settings. We can now treat animals with a drug and then slice and dice them and heat it up and find out what happened, basically what are the drug targets, but also what is the melting behavior like in living tissue. And finally, together with Bernard Küster, we did this across the tree of life for many different organisms and found out a lot of aspects of how thermostability links to protein function. I want to talk about this particular work on prokaryotes. This is our latest and I would say biggest work in the lab, which was accepted in Nature just recently. And so what we set out to do here, and this was work done by a really brilliant postdoc in the lab, Andre Mateos, we set out to combine reverse genetics with thermal protein profiling to understand how the E. coli protein network is wired and basically creates a functional proteome. And the idea and the experimental approach is that we selected 121 mutants from single gene deletion library in E. coli, which were involved in diverse processes. So we wanted to the protein perturbed in many different ways, including also genes of unknown function. We had 2 independent clones for each mutant, and we used at that time TMT11. Now we're switching to TMT16, but basically we multiplexed 11 mutants in each condition, and each mutant, the 2 different clones were multiplexed in this way that they always appeared with different mutants, and we calculated the changes in stability and abundance against mutant. A lot of experiments. I think this was around 90 days of instrument time in total. And in terms of what we can get out of these experiments, when we compare control versus knockout, so changes in thermostability, stabilization, destabilization, but also changes in protein abundance, and of course, combinations of the 2. We can calculate the abundance caused by using the 2 lowest temperatures, where we know that stability doesn't change. Can correct for that and see if we have on top of that stability changes. And we can convert these values to z-scores, calculate Q values using lemma, and finally end up with a number of hits. And so in total, we profiled 1,764 proteins for which we got abundance and thermostability scores, and 1,215 of them turn out to be hits in at least 1 mutant. So we basically perturbed quite a large subset of the E. coli proteome. And roughly similar numbers, 850 or so, were hits in the abundance of thermostability. And what you can already see from the heat maps on your right is that basically there's very little correlation between abundance and stability, which is an excellent thing because it basically tells you that stability brings something to the table which abundance does not. And indeed, looking at that in more detail, you have another number of these clusters, small clusters in the thermostability heat map. And if you look at them more directly, you see that, for instance, here, this is a nice illustration. So these 3 mutants are iron sulfur cluster by synthesis mutants, so they cannot synthesize iron clusters. And the iron sulfur cluster binding proteins get very, very strongly destabilized in these particular mutants. So we get actual functional formation. This is not reflected in abundance. Now, a very exciting point of the study was regarding essential proteins. So it's a number of essential proteins in E. coli which cannot be knocked out, and so they're very difficult to study using genetics techniques. And so what we found out is that in our screen, the essential proteins were hits as often as non-essential proteins. But excitingly, there were very, very rarely abundance hits, and there were very, very often hits in protein thermostability. Now, these proteins are more abundant in general than non-essential proteins, and as already said, they're hits in thermostability typically. So this made us think whether this change in thermostability of an essential protein actually implies or tells us about how important it becomes for the cell. And in order to validate that, we used the recently developed CRISPR-I technique to basically downregulate the levels of essential proteins in mutants where they were changing in thermostability and in mutants where they were not changing in thermostability, in order to basically see whether those mutants where they were changing thermostability became less viable. And this turned out to be the case, basically, where the mutants where the essential proteins were changing in thermostability became more sensitive to downregulation of the specific essential proteins. Another very exciting thing from the study is, of course, that we have a lot of data points for each protein. We have 1,210 data points for all the proteins for which we record measurements across all the different temperatures and all the different mutants. And so we can now take these long vectors and correlate with each other and basically see which proteins correlate with each other and then see if they're functionally related. Below is an example for RPOB and RPOC, 2 proteins which are part of the E. coli RNA polymerase. And you see that they correlate very nicely. These are two essential proteins. You also see that they correlate due to changes in their thermostability and not due to changes in their abundance. And if we do that systematically, we see this formation of a number of clusters. And when we relate that to what is known about E. coli, we see that very nicely we recapitulate operons, protein complexes, and metabolic pathways, and also protein-protein interactions. And this is, of course, for the known things, but there's a number of also orphan proteins still present in E. coli, and we could find interesting functional annotations for these proteins, which have had no functional annotation before. Furthermore, we looked at metabolism. This is the central carbon metabolism. Again, if you make these correlations, you will see a nice formation of 3 distinct clusters corresponding to different parts of the carbon metabolism. And we also wanted to see whether the thermostability related actually to the levels of the respective metabolites. So we did targeted metabolomics for 6 metabolites of the central carbon metabolisms across 19 mutants. And then we looked how that related to the protein stability of the proteins of which these metabolites are substrates. And indeed, we observed nice correlations between the thermostability of proteins and the levels of metabolites that we measured. But this correlation was pretty much completely absent when looking at abundance changes in these proteins. And this was a trend which we recapitulated pretty much in all the measurements, basically telling us that metabolite levels correlate to thermostability and that we can get information on the metabolic state of a living cell by doing this type of experiments. And then finally, we also had a genetic screen for different phenotypic screen of all these mutants. So we knew in which different stress conditions different mutants exhibit growth phenotypes when they change in growth. And we basically asked ourselves, so do these changes on the proteome level, how do they relate to changes to the number of phenotypes that particular mutants have? And there was actually quite a nice relation. So basically, if you take a specific E. coli mutant and you see how many growth phenotypes it has across thousands of conditions, you look at that number X, and then you see, and then you look at how many proteins are perturbed in abundance and stability in these mutants, you see that these two measures are actually related. And so the more a mutant, specific mutant is perturbed on the proteome level, the more phenotypes it will have, which then immediately suggests that the changes that we observe on the proteome level are directly related to the different phenotypes. And that we can also infer things about the specific mutants and the different phenotypes, which are not directly related to the gene which is being deleted. Here's just one trivial example where the gene YBAB was deleted, and this led to sensitivity under UV ultraviolet conditions, which made people actually postulate that this protein plays a role in UV radiation. But what we could show in this rather simplistic case, that this was due to a different protein, REC-R, which actually showed the correlation with the UV phenotypes, and which is located right next to YBAB, which was also getting down-regulated. And we could show by rescue experiments that adding REC-R but not YBAB actually rescues the phenotype in this particular mutant. So in order to recapitulate this part of the talk, we performed thermal protein profiling of E. coli after more than 100 genetic perturbations. We observed that levels of essential proteins are high and rarely modulated. So these proteins are difficult to study by proteomics and genetics, but thermostability of essential proteins was affected and reflected their function. And protein correlation recapitulates known biology, but also tells us a lot about orphan gene and assigns putative functions to these genes and proteins. And finally, genetic perturbations lead to changes in abundance and thermostability that are related to their function. And thermostability of enzymes reflect metabolite levels, and protein abundance and thermostability can explain growth phenotypes beyond the deleted gene. So this will be very valuable data set for basically studying E. coli, but we also can now extend this approach to less characterized organisms, where this will be very exciting because we'll find out many more things that were not known before. So this brings me to the second part of the talk, where I just like to point out that, of course, such huge studies require really robust and high quality raw file processing and raw mass spectrometry data processing. So we need something which is flexible, adaptable, broadly usable to meet the needs of large-scale proteomics. It needs to be easily usable for us in the lab, fast processing, automated to reduce time, demand on repetitive tasks, and needs to be easily integrated into existing pipelines. So we had been working for many years, also when I worked at Cellsum, at software for analysis of tandem mass specs, and we were using the software until quite recently called IsoBarQuant, where we incorporated all the different tricks that we learned over the years of how best to work with tandem mass specs. Apologies. And then we were contacted by Biognosys, and actually together with them, we've been working, they've been helping us to adapt SpectroMine to deal in the best possible way with tandem mass spec based quantification. This was really fantastic because they've been incredibly helpful, incredibly responsive, and they incorporated all the key elements of all the things that we learned over the years into SpectroMine, which led to really excellent results in terms of tandem mass spec quantification. And the fantastic thing about SpectroMine, there are several ones. It's very, very easy to use. It's scriptable from the command line, so we could very, very easily incorporate it into our pipeline. It's incredibly fast, so it's 3 times faster than the tools we used before, and it's also a single step thing. So it again speeds up the processing pipeline and requires much less time from the people in order to analyze this type of data. And as I said, we spend a lot of time just also make sure to validate that we're getting the same quality of results that we were used to with the previous software, and we are getting that, but we're getting it now much faster and in a much easier way, which is much more useful to us. So I can only highly recommend that. And this is basically just showing that we're getting similar results. We get slightly, in terms of overall protein identification and quantification accuracy, we actually get now slightly better with SpectroMine. And so I think this has been really a success story, and we'll use it very extensively going forward. With that, I would like to thank you for inviting me, and I would like to thank the members of my group, close collaborators, Nasser Stipas, Martin Beck, Per Borg, Wolfgang Huber, Markus Panchev, and I'd like to thank you for your attention. Thank you very much.
Thank you very much, Misha, for this very interesting presentation, giving insights into the structural proteome of E. coli. Again, to the audience, please remember to ask your questions in the Q&A box in Zoom, such that we can answer them in the end after both presentations. Please don't forget that. And with this, I would like to hand over to Irving, our second speaker of today's user seminar. Irving's background is in biochemistry and molecular biology. He did his PhD modeling cell behavior, and he was recently appointed associate professor in single-cell proteomics in disease biology at DTU in Denmark. His research focuses on normal and malignant hematopoiesis, and he has spent the last 3 years optimizing a single-cell proteomics workflow to be used for deciphering cellular heterogeneity. He's also the head of the DTU Proteomics Core, a facility that is specialized in high-sensitivity proteomics workflows. And with this, I would like to hand over to Irving for the second part of today's presentation.
Thank you, Lucas, for that kind introduction, and also, of course, for the opportunity to present here. And thanks to the whole Biognosys team for the tremendous support that we've had while trying to implement SpectroMine into our single-cell workflows. So today, I just wanted to give you a little story on how we're now doing quantitative single-cell proteomics. Basically, give a brief background, give a brief experimental overview of the workflow, how we're using SpectroMine to optimize our search strategies, showcase some few results, and finish with a few future perspectives. So Of course, a question to start with is why should we care about single cell technologies? And I think we can go over this relatively quickly because there has simply been a shift in modern biology in terms of trying to discover cell heterogeneity at single cell level rather than just trying to deconvolute from a complex mixture. So not only in the skin or bacterial fungal colonies where we know different cells are interacting, but in the case of my research, especially the blood and immune system, where we know we have this hierarchical nature of different cell types interacting and differentiating to give rise to the entire blood system itself. And then, of course, we have a strong focus on figuring out how this goes wrong in a cancer setting. And we have a special focus on the cancer stem cells that we know evade chemotherapy and can cause relapse after treatment in the patient. And really, if we want to get to the bottom of the signaling of these individual cell types, we simply have to look at them at single cell level. So all the work I'm presenting today is based on this A227 culture system, which maintains the leukemic hierarchy even in vitro by culturing it under very closely controlled conditions. So for those of you less familiar, the AML hierarchy really consists of leukemia stem cells at the apex, and the arrow here indicates that these can self-renew, i.e. make more leukemia stem cells, and they differentiate into leukemic progenitors and subsequently into leukemic blasts. And it's these blasts that make up the bulk of a leukemia tumor and end up causing the bone marrow failure that patients generally present with in the clinic. So Eric Leckman in the John Dick lab managed to find this relapse AML sample and figure out how to culture it in vitro while still maintaining the hierarchy. And it's just important to understand how this works for me to continue on with the data that we generated. So this plot is showing the CD34, CD38 FACS plot of this cell culture system. And we can see that 90% of the cells are located over here, and these are the blasts that are terminally differentiated. If we take these 34 positive, 38 negative cells, put them in culture, 7 days later, we see that the entire hierarchy has been recapitulated. If, on the other hand, we take the progenitors and look at the cells 7 days later, we actually only see the blasts and a few remaining progenitors having been recreated, therefore really showing that it's only the LSCs that are able to self-renew and recapitulate the entire hierarchy. The good thing of this cell system for us in terms of developing single cell is that this system represents a heterogeneous cell population containing different cell differentiation stages that we should therefore be able to pick apart by using our quantitative workflows. So how do we do single cell proteomics? So we, of course, have learned from previous efforts and took the approach of Bogdan Budnik and Nikolai Slavov, published a few years ago, and then have made some upgrades in terms of now using the TMT Pro. So we have 16-plex. The main reason for going down this road for us is, one, the multiplexing capability that we can run multiple cells at the same time, but also to allow the inclusion of a booster channel, in our case 200 cells, which just adds more ions to the mix, because as you can see here, one cell is roughly equal to 200 picograms, so it's really pushing the boundaries of sensitivity in terms of using standard MS workflows. So by individually labeling single cells and a booster channel and combining them, we run them on an Orbitrap Explorers 480, combined with a FAMES Pro ion mobility device and the EZ1200, and we developed a pipeline called Scepter, which we can use to visualize the data, where we can show cell clustering, how many proteins we identify per cell, and visualize individual protein expression levels on the embedding of choice. In terms of practical workflows, this is an overview of how we prep the cells. So we use FACS ARIA2 sorter in this case, but any cell sorter that can be combined with a 384-well plate collection device will work. I don't have time to go into depth, but we actually now separate the booster and the single cell plates for various reasons. And basically we sort the single cells into 1 microliter of lysis buffer. Then we boil the cells to lyse them and denature them. And then we use this Dispendix I.1, which is a non-contact liquid dispenser, to add the trypsin, the TMT reagents, and basically do the entire digestion and labeling reaction in a very small volume. Then we use this OpenTrons robot that many of you will have heard to actually pool the samples. So this is just a small video showing how it's done. So we have our booster sample in here. So we're just priming the tips with that booster sample because we collect everything in the 96-well plate. And then now we're starting to collect all the single cells from the 384-well plate that we used for the prep. And this turns out to be a really efficient way of preparing many plates in a reproducible manner, and therefore get reproducible results at the end when we're looking at the data. So in terms of using the data that comes out, because we're still using very low amounts of protein or peptides on the column, so we need a very sensitive method to detect these. So Lucas kindly reached out and asked whether SpectroMine would be of use here, so we figured why not test it. So if we compare to another vendor that we've been using before, and we just search our files, in this case 24 runs against a FASTA file, we can see that just by using SpectroMine, we're already gaining up to about 1,300 proteins from 1,250 proteins per injection on average. If we then add the library function that's included in SpectroMine, we actually gain another 30, 40 proteins per injection on average, as shown here. And then what we thought, how about we add a sample specific, but a higher load library, basically using the same cells, but just in bulk, fractionating with high pH and running on the same MS settings, we actually do gain a tiny bit more, 10, 20 proteins per injection, just to add more data or more knowledge from the data that we generated. Of course, if we just use this high pH library, we don't get nearly as much coverage, but this is to be expected, and just to showcase that you do need to be a bit careful about what spectral library search strategy you take. So how does this translate to actual results? I mean, before I talk about the results we get, we need to touch a little bit on how we measure these samples, because as I mentioned, we have a very low amount of sample loaded on columns. So if in this cartoon, we're just depicting the actual ion signal from a sample. If we use a 300 millisecond injection time, we're capturing a portion of it, and we increase that portion proportionally if we almost double that to 500 milliseconds. But we didn't really know what would lead to an accurate result. So we did a triplicate injection in terms of testing 4 different settings, 150 milliseconds all the way through a full second for MS2 collection time, and then plot how does the signal to noise change. And we can see that the longer injection times lead to a better signal to noise in the single cell channels. And that also translates to an improved CV value between measurements, i.e. are we measuring the same protein levels consistently? And we can clearly see that the higher the injection time, the more accurate our measurements become. And as summarized in this table, we basically see that from 300 on, we're starting to reach a workable number with a CV less than 20%. And at the 500 milliseconds, we're actually measuring half of our proteins with a CV of less than 20%. But to test this more thoroughly, we figured let's try both and basically measure 24 injections each, both at 300 and 500 milliseconds, i.e. 672 single cells in total. So these were the cells that we sorted, again, plotted under CD34, CD38. So we had a lot of LSCs, a lot of progenitors, and a lot of blasts. And then basically plotted here in this table, which is not visualizing fully properly, but basically in this column here, we have the per cell number of proteins identified in the 300 milliseconds with FASTA, 300 millisecond with Spectral library, and then the 500 with FASTA and Spectral library. And we can see that we get a slight boost every time by using Spectral library searching. And compared to other vendors, we see that we are clearly outperforming them and basically getting free extra knowledge just by using SpectroMine. How does this look in terms of biology? So if we use our Scepter pipeline, which I must stress has been really pioneered by a great PhD student in the lab, Benjamin Furtwängler, basically it's our computational pipeline to visualize and extract meaning from this. So we're just plotting the 2 different MS settings, and then whether we use the FASTA file or Spectral library for searching. And we can see that no matter what settings we used, we get this clear separation away of the blast from the LSE progenitor. And that's what we know because the blasts are fully differentiated. And actually, in fact, we even managed to separate the LSE progenitor to quite some extent in the 300 millisecond and even a bit more in the 500 millisecond setting, showing that our data is actually capturing the phenotypes of these cells that we measured. So here we can just plot how many proteins were identified per cell just for visual interpretation. And what's really nice about the latest version of Scepter is that we can now plot the FACS data back on top of this. So if you look at the CD34, we can see that it's indeed the progenitors and the LSCs that are CD34 positive, which is exactly confirming what we knew from our FACS data. But the fact that our protein level clustering based on the MS data is able to cluster these cells correctly that correlates with the FACS data was very promising. And it's the same for CD38, that we have our progenitors being 38 positive, our LSCs 38 negative, and our blasts are both 38 positive and negative, as we knew from our FACS data. So this looked very promising because it suggests that just using SpectroMine, either with a FASTA file or Spectral library, is not changing the quantitative accuracy of the data, and is therefore a good measure to just boost your per cell protein number quite significantly. What else can we do with Scepter? I mean, this is just showing based on the 500 millisecond data and the Spectral library search. We can ask it, give us all the proteins that are cell specific, for example, LSC progenitor versus blast. And we can pinpoint those that seem to be LSC progenitor specific. We can then plot these proteins based on the UMAP layout that was created by Scepter using the quantitative proteomics data. And here we can again see these three clusters of the cell types, and we can see that both lysozyme and NPO seem to be a blast-specific protein in this case. We can ask Scepter to do unsupervised clustering, which is very relevant in case you want to find new cell types, because we know blast, LSC, and progenitors exist, but there's probably several intermediate states of differentiation that we could use the single-cell proteomics data to figure out. And indeed, if we ask Scepter to do the clustering for us, it's actually finding 5 different clusters. And then we can again extract, okay, what are the proteins that are highly upregulated in cluster 1 versus the rest, and then we can do that for all the clusters. We can visualize this on a heat map just to figure out, okay, which proteins are truly seeming to be relevant here. And it really just allows you to extract meaning from your data. So just to summarize, I mean, the SpectroMine 2.0 seems very well suited towards identifying MS2 spectra from low abundance TMT samples. I mean, we definitely get 10% more IDs. So I mean, instead of getting 1,250 proteins per sample, we're now getting 1,350. Might not sound like a lot, but if you translate that to peptides, we're actually getting, instead of 5,000 peptides, we're getting 55,000 peptides per run, which simply adds that quantitative accuracy if you have more peptides per protein to quantify the TMT signal from. Similar to Misha before, we found that the searches are extremely fast. Really takes a few hours versus 1 to 1.5 days for 24 single cell runs. When we add the library search function, we get about 3% more proteins and peptides. Importantly, this also reduces the number of missing values between single cells. So we're down to about 56% across 295 cells. And the clustering, as I showed before, seems very similar. So it doesn't look like there's any quality loss in terms of signal accuracy. Our results do indicate that the Spectral library performs best when taken from the exact same or mimicking samples. So we haven't yet, at least, been able to leverage significantly more IDs by deploying higher load samples. But I guess future AI perspectives might change this. But definitely, I think it's worth concluding that exploring search engine strategies for your single cell mass spec work is worth your time. In terms of future perspectives, I mean, I think we can now safely say, and as shown by others, that global single cell mass spec seems to work. We've been working really hard the past few months on getting targeted single cell mass spec to work in order to be able to do consistent cell-to-cell quantitation of specific protein panels, either using short quant or single cell tomato. So I recommend to watch the space. I've now basically, we've done a lot of testing on chromatography, but we'll continue to do so because there is still a lot of gain to be had here. We're currently running about 120 cells per day per instrument, but of course, to do really big studies, we should improve this. So if we decrease our gradient to a 2-hour versus 3-hour, we're getting only about 15% less protein IDs, 20% less peptide IDs. So this is a strategy, but for sure, using maybe ultra low flow or picoflow, we might be able to decrease the runtime even more. And we're currently investigating MS3 level quant with real-time search on the eclipse and how that impacts the cell heterogeneity. With that, I would really like to thank the people involved, especially Bo, who's been very supportive during my postdoc getting this method developed. Benjamin, who's behind the Scepter development and experimental development, and Neil was really integrative for the experiments and setting up the buffers and the volumes, etc. Colleen and Despina for cell sorting. Ulrich and Simonis at DTU who've been seminal for getting this to work, the entire DTU proteomics core. Various people at Thermo Fisher who've helped setting everything up, other technology partners, and of course the funding. Thank you for your attention. I'd be happy to take questions.
Thank you. Thank you very much, Irving and Misha, for these wonderful presentations. Again, I kindly ask the audience to ask questions using the Zoom functionality, and we already have a few coming in, so I will directly jump into it. One question asking Misha, great data and promising approach. How do you handle the DDA reproducibility problems, measuring many TMT pools, use of libraries, question mark? So I guess he's referring to the batches in these large-scale TMT experiments.
Yeah, that's an excellent question. I mean, it depends on what you want to get out. For this particular, for the large E. coli study, there was not such a huge problem because the proteome is relatively low complexity, and we additionally also fractionated the samples to really also minimize the ratio compression. So we're getting excellent coverage in most experiments. So of course there are some proteins, really low abundant proteins, which in some experiments you get, in some others you don't, and there was that level of variability. But the bulk of it, the 1700 proteins, were really reliably quantified anyway across all the different conditions. So that was not really an issue.
Do you use any criteria that it has to be observed in so and so many batches to kind of pre-filter your data, or not necessarily?
Again, it depended a bit here. So there were different aspects of the study. For instance, when you look at how in a particular mutant the thermal stability abundance changes, we required that the protein would be quantified in both clones and also and that we get those values. But then when looking at individual mutants, it was not required, that this protein was also identified in all the other 100 mutants. So that was necessary. However, for the correlation analysis of proteins across the different mutants, there we had the cutoff. So the protein had to be seen in at least 60% of all the mutants so that the vector would be sufficiently long. And then we used it for the correlation analysis.
Mm-hmm. Thank you, Misha. There's another question for Irving. Somebody asks whether you can comment on the strategy to inject the samples from plate onto column, as I guess, despite the fact that you're using a booster channel, there are probably still losses in the auto sampler. So maybe you can comment on that, Irving?
Yes, absolutely. So I think it's actually it works quite well just going from a 96-well plate directly into your auto sampler. So as mentioned, we basically have the booster sample in the 96-well plate, and then we collect all the single cells into those same wells. We speed vac it, and then we resuspend the sample in 8 microliter of 0.5% TFA to directly put that plate into the auto sampler. So this also means that you can run for a week or two from the same plate. And because we use the Eppendorf low bind plastic, we don't really seem to find much sample loss at that point, at least.
Mm-hmm. Okay, thank you very much. somebody asks to both speakers, sorry if I missed it, but were you using an MS2 or an SPS MS3 method in the data you have presented?
So we usually use MS2, personal opinion, because we usually find that it's more robust quantification, and we fractionate deeply enough, so we fractionate quite deeply, so we don't have much ratio compression. And usually in this type of experiments that I showed here, where we look at any change in terms of stability and abundance, it's not the most essential thing for us if the fold change is 2.3 or 2.1. It's more detecting that there is a change, and so there we're fine. In some specific, when we combine TMT with turnover measurements, which we also sometimes do, there we use SPS MS3 basically to get as accurate as possible. So there for us it makes sense.
Oh, it makes sense. And Irving?
Yeah, so we indeed so far have been using MS2, but because we combine it with the FAMES Pro and multi-CV gas phase fractionation, our co-isolation interference is generally around 10%, so quite low. We have tested on an old fusion with the SPS MS3, but we take too significant a loss in terms of ID rates. We have tested together with San Jose on the Eclipse, and we'll be taking delivery of such an instrument very soon. So of course we will develop that method much further, because I think the real-time search will mean that we don't lose the ID rate, but indeed should get the benefit of the MS3 level quant.
Okay, thank you very much. I have another question to Misha. So you did not touch too much on the mechanisms by which proteins can be thermally destabilized or stabilized. I guess you have been looking at this in past publications, but still regarding your presentation, what do you think are the main mechanisms, and particularly what do you think are the differences between a prokaryote such as E. coli compared to a eukaryote such as human cells, for instance, in terms of mechanisms?
That's an interesting question. Regarding differences between prokaryotes and eukaryotes, not sure I can give a good answer whether we observe any differences due to what proteins are stabilized. Proteins can change their stability due to a number of things, and that's also an really exciting part of that field. So of course, it can be protein metabolite interactions. When you see an enzyme changing in stability, it's typically due to that. Of course, it has to be validated. For instance, we've before observed stabilization of the RNA polymerase, and we showed in vitro that this is due to interaction with DNA. We've also seen cases where we knock out complex members and other complex members get destabilized. So it's a learning process. So in many things you can actually deduce quite easily what the change is due to. In other cases, you need to find out what causes the change. Like for instance, with the case of RNA polymerase, and now we know that if it changes in thermal stability, this is most likely due to that. For a number of other proteins with function which we haven't studied much, we still don't know what the changes, what the reason for the changes is. And this is an exciting part because, of course, when you follow up experiments to understand what changes in this protein function modulate the stability changes. So that's an ongoing thing.
Mm-hmm. Okay, thanks very much. A question to Irving. So the person asks or says that it's known that in HeLa experiments or under optimal conditions, roughly 50% of MS2 spectra can be annotated. That's really sort of an upper bound in these kind of experiments. So in these single cell experiments, do you know roughly what is the percentage of spectra that can be annotated?
Yes, great question. I mean, we're at about 20 to 25%, depending whether we use the 300 millisecond injection time or the 500 millisecond injection time. So for sure, we are lower than a standard HeLa experiment, but yeah, we are still at a level where it's workable. So we're not wasting too many scans on things that weren't identified to begin with. So it's also a matter of carefully selecting your intensity thresholds in terms of not selecting the really low abundant that you would never be able to identify in the first place.
Mm-hmm. Yeah, that's very interesting. Maybe I have a second last question here. that would be again to Irving. So what do you think, where will single-cell proteomics start to make an impact first on the application side, let's say?
You mean disease area or?
For instance, yeah.
Yeah, I mean, I think we will learn a lot about cellular hierarchies and the differentiation trajectories. But indeed, in order to get there, we need to boost the number of cells. I mean, we're all very proud when we can do 1,000 cells and 1,000 proteins per cell. But we're probably looking at tens of thousands of cells that we ideally need to be interrogating to truly get meaning, because the cell frequencies are quite low. Like, for example, in this A227 sample, the LSCs only make up 3% of the cells, meaning you need to run a lot of cells if you want to interrogate enough of them to truly understand the cell signaling in them. But that said, I mean, we are finding cell type specific proteins already. I think in terms of drug treatments, it will be very interesting to see why certain cells die during drug treatment and why others don't. And that would be an interesting avenue to take with single cell proteomics.
Mm-hmm. Okay, very interesting. And then maybe my last question. So to bring these two topics together, maybe a crazy question, but have you ever thought about doing a single-cell thermal proteome profiling?
Yeah, I have actually, but it's obviously quite challenging for. In its current form, it will be tricky because, like you heat different aliquots of cells to different temperatures, so that's where it kind of becomes a no-go. But you could assume scenarios where heating it to a single temperature potentially, where particular protein changes in thermal stability, and assuming that the level of this protein does not vary too much across the single cells, potentially that could work, but that's a bit far-fetched.
And Irving, do you want to add something to that?
Yeah, I mean, I think it's a matter of trying. Since we use a PCR machine to boil the samples anyway, I guess we could vary the temperature of plates individually and then run a few plates. I mean, I'd be happy to talk at some point, Misha, if that would be of interest. Yeah. It would be very cool, absolutely, especially also with the drug treatment remark that I made. It would be very relevant.
I agree.
Okay, yeah, thank you again very much to the speakers, not only for the wonderful presentations, but also for the interactions we had in the past months. So this was very insightful for us, and yeah, I hope we can keep this going. So thank you again, and thanks to the audience for your interest. Bye, everybody.